Hello everyone. In this lecture, we'll talk about the multiplication of viruses. Viruses multiply only in living cells. The process of intracellular viral multiplication is called self-replication, consisting of the synthesis of proteins and nucleic acids of viruses and assembly into new infectious particles. Although the details vary from group to group, the general outline of the replication cycles is similar. A virus multiplication cycle is typically divided into the following phases: adsorption to the host cell, penetration or entry into the cell, encoding to release the genome, biosynthesis, the process of virion component production, and finally, the progeny virions are assembled from the replicated genetic material and newly made capsid proteins, get maturation, and release from the cell. The first step in viral infection is adsorption or attachment, interaction of a virion with a specific receptor site on the surface of a cell. The specificity of attachment determines the host range or tissue tropism of the virus. This cartoon shows the absorption of a naked virus. VAP, the proteins on the surface of the virion, attach to the specific receptor proteins on the cell surface. This cartoon shows the adsorption of an enveloped virus. Spikes on the surface of the virion attach to the specific receptors on the cell surface. After bending, the virus particle is taken up inside the cell. This step is referred to as penetration or engulfment. There are three main ways. Endocytosis means engulfment of the virion by host cell. Most naked viruses enter the cell in this way. Fusion of virus envelope with host cell membrane. Most enveloped viruses enter the cell in this way. Direct entry, which is a translocation of the virion across the plasma membrane. Some naked viruses enter the cell in this way. In some systems, this is accomplished by receptor-mediated endocytosis, which uptake of the ingested virus particles within endosomes. There is fusion of the virion envelope with the plasma membrane of the cell for an enveloped virus. Those systems involve the interaction of a viral fusion protein with a second cellular receptor or co-receptor. Encoding occurs concomitantly with or shortly after penetration. The viral nucleic acid is released from the capsid and is available for transcription, translation, or replication within the cell to form new progeny virions. A low pH within the vesicle favors encoding. Lysosomal enzymes in the cells are involved in this process. The biosynthesis of viral components include viral gene expression, synthesis of viral proteins, and genome replication. Several processes may occur before new viral particles can be assembled. First, transcription of viral nucleic acid to mRNA. This process is also called early transcription. Second, translation of mRNA to proteins. The proteins are mainly non-structural proteins, acting as enzymes, and they are necessary for viral replication. This process is also known as early translation. Third, replication of viral nucleic acid. Fourth, transcription of viral nucleic acid to mRNA, also known as late transcription. And fifth, translation of late mRNA to viral proteins, which are structural proteins, and this process is also called late translation. Various classes of viruses use different pathways to synthesize the mRNA depending on the structure of the viral nucleic acid. We will talk about four common types of biosynthesis. The dsDNA virus transcription occurs in the nucleus, using the host cell DNA-dependent RNA polymerase to synthesize the mRNA. The replication of the double-stranded DNA virus can be divided into two stages. In the early stage, the genomes of most DNA viruses are transcribed by the host DNA-dependent RNA polymerase, RNA polymerase II, in the nucleus to yield the viral mRNA, which are exported to the cytoplasm for translation. The early proteins are non-structural proteins. The major components are DNA polymerase and a series of enzymes which regulate the gene transcription and inhibit metabolism of host cells, and provide the conditions for the virus to replicate. In the late stage, the viral double-stranded DNA was replicated by the enzyme produced in the early stage, and mRNA transcribed from the viral DNA can enter the cytoplasm and be translated to the viral structure protein, including the capsid protein. Most RNA viruses undergo their entire replicative cycle in the cytoplasm. We'll talk about three types of RNA viruses: positive single-stranded RNA viruses, negative single-stranded RNA viruses, and retroviruses. The genome of the positive single-stranded RNA viruses can not only be used as a template to replicate the RNA of the progeny viruses, but also act as mRNA, carrying genetic information for the synthesis of both structural and non-structural proteins. When the virus enters the cell, the viral RNA binds to ribosomes, which acts as mRNA and translates early proteins, including RNA polymerase and protease. Using the polymerase enzyme, negative strand RNA is used as a template for the synthesis of complementary strand. A temporary double-stranded RNA is formed, which is called the replicative intermediate. The negative strand RNA can be used as a template to make more progeny positive strand RNA, and the positive strand RNA of the replicative intermediate acts as mRNA for translation into more viral proteins, mainly new capsid proteins. The negative single-stranded RNA has negative polarity and is complementary to mRNA. The genome of negative single-stranded RNA viruses can be used as a template but cannot act as mRNA. Because the host cell does not have an RNA polymerase capable of using RNA as a template, the virus carries its own RNA-dependent polymerase. Using the RNA-dependent polymerase, negative strand RNA is used as a template for the synthesis of complementary strand. A temporary double-stranded RNA is formed, which is called the replicative intermediate. The positive RNA of the intermediate acts as mRNA, translating into more viral proteins, including non-structural proteins such as RNA-dependent polymerase, and structural protein such as capsid protein. The positive RNA of the intermediate can also be used as template to make more progeny negative strand RNA. Retroviruses are the only family of viruses that contain reverse transcriptase, an enzyme with RNA-dependent DNA polymerase activity. Retroviruses have two copies of single-stranded RNA. Complementary strand of DNA is synthesized using RNA as a template by RNA-dependent DNA polymerase, and a hybrid of RNA and DNA is formed. In addition to RNA-dependent DNA polymerase activity, the reverse transcriptase has RNase-H activity, which is able to digest RNA from a DNA-RNA hybrid. The resulting single-stranded DNA is converted into double-stranded DNA by the action of the DNA-dependent DNA polymerase activity of the same reverse transcriptase. Double-stranded DNA migrates to the nucleus where it integrates into the host cell DNA. Integration is mediated by a virus-encoded integrase. The integrated viral DNA is called provirus. As the DNA of the cell is replicated during cell growth, the viral DNA is also replicated. This DNA copy is then transcribed into viral mRNA by the regular host cell RNA polymerase and translated into viral proteins. HIV synthesizes large polyprotein precursors that are processed and cleaved to generate the final gene products by the viral protease. The final stage of replication cycle is assembly, maturation, and release. The progeny particles are assembled by packaging the viral nucleic acid within the capsid proteins. Helical nucleic capsids are assembled by adding protein subunits to the RNA genome to form a helix. Icosahedral capsids are pre-assembled, and the nucleic acid genomes are complexed with condensing proteins. DNA viruses are assembled in the nucleus, except the pox virus. Most RNA viruses are assembled in the cytoplasm. Maturation is the process by which viruses become infectious. Virus particles are released from the cell by either of the two processes. One is the rupture of the cell membrane and release of the mature particles. This usually occurs with naked viruses. The other, which occurs with enveloped viruses, is release of virus by budding through the outer cell membrane. Enveloped viruses are not infectious until they have acquired their envelopes. The two cartoons show the release of the naked viruses and enveloped viruses. This electron microscope image shows the budding of the viruses. Let's have a look at the whole replication cycle of viruses. Adsorption or attachment of the virus to the host cell, penetration, uncoating, biosynthesis, including replication of viral genome and translation of viral proteins, and finally assembly, maturation, and release. After one replication cycle, a virus could produce a large number of progeny viruses, which infect other host cells to start the new replication cycle. Okay, that's all for the multiplication of viruses. Thank you for watching and see you next time.